Okay, to remind you, so when we're going through the myosin and actin interactions and contraction, there's a lot of stuff going on, but the key to switching back and forth between the power stroke that's moving actin and the recovery stroke, detachment and going back to the cocked position, the keys here are what the phosphate is doing. When a phosphate is attached, so when a phosphate is there, ATP has bound, the phosphate has separated out. At that point, it goes to the cocked position. In this schematic, I've just put a couple of plus charges up here to give you the idea that, sure, a phosphate would pull that up around into the cocked position. Okay. The actual charge distribution is different, but the schematic gives you the sense of, okay, if we put a -2 charge on there, it's going to change the shape. Okay. And binding to actin then triggers that phosphate to pop free. So binding to actin means that phosphate no longer fits in its little binding pocket. Once actin binds, changes the shape of myosin, the head, phosphate pops free, and without it, you've got nothing holding the myosin head in the cocked position. And so the myosin head moves, that's the power stroke, and it moves actin, okay, in that direction. So phosphate comes off as it's leaving, that's the power stroke, and then bind a new ATP and separate out the phosphate, that's the recovery. Okay, so when you're practicing this, key is to think first about what the phosphate does, and then think, okay, how are you going to get a phosphate back on? Well, once the phosphate is popped free, you're stuck, attached to actin, you need a new ATP. That new ATP binds, and once it separates into an ADP and a phosphate, that will allow the myosin head to go back to the cocked position. Bind to actin, off pops the phosphate, and the power stroke. Okay, so if we change some part of it, so if, for example, we make the binding pocket have a little higher affinity to phosphate here, how do we change contraction? So if this binding pocket attracts phosphate a little more strongly, how do we change contraction? So tell your neighbor what's going to be different. Okay, so higher affinity. It sticks a little bit longer. You're going to slow that down. And in fact, there are myosins. You have multiple myosin genes that are functioning in different ways, even in different parts of your body. And across animals, there are myosins that cycle fast, which means fast contraction, but a lot of ATP. They're going through ATP at a very high rate. So they'll exhaust fatigue much more quickly. And then there are myosins that cycle really slowly, which gives you a very slow contraction, but they don't fatigue as quickly, and they don't use ATP as quickly. So a trade-off between speed and expense. The faster you want that muscle to contract, the more expensive it's going to be. Okay, so here's a textbook figure, not very useful because it's so complicated, but it has everything in it. Here's a textbook figure showing how an action potential coming from a motor neuron down to what's called a motor end plate, but really these are just synapses. They're simply synapses of a motor neuron on a skeletal muscle cell. Okay? So motor end plate, but actually synapses, these little blue spots here. If you look at them close up, they look like a typical axon terminal, okay, with the action potential coming down, neurotransmitter release, and then events here. That's what was in the video you guys have watched. So if we look at a synapse of a motor neuron on a skeletal muscle cell, we have an action potential coming down, just like before. Voltage-gated sodium channels opening up at the first half of that action potential. Sodium gets in, and we open voltage-gated calcium channels down at the tip of the axon, at the axon terminal. Calcium binds to a calcium binding protein. That calcium binding protein causes the vesicles of a neurotransmitter to fuse with the membrane, and it releases the neurotransmitter. In skeletal muscle cells, the neurotransmitter is acetylcholine, represented by this little triangle, or ACH. And what would you guess? Is this likely to be a metabotropic or an ionotropic receptor? Okay. Do you care about speed? Okay, absolutely. So, ionotropic receptor. So, the acetylcholine receptor is an ion channel. It's an ionotropic sodium channel. Okay, so that brings sodium into the cell. Skeletal muscle cells can produce action potentials, just like a neuron, exactly the same set of proteins. Okay, so skeletal muscle cells can produce action potentials. So the sodium coming in will open voltage-gated sodium channels. I'm not showing voltage-gated potassium channels for the second half of the action potential, but you know they're there. And sodium gets into the cell. This is messy, but hopefully you've gone through the video, and so you recognize the parts. Here's the surface of our muscle cell, a muscle fiber. Here's the membrane. This is a T-tubule, transverse tubule. Just means when people were first slicing up these muscle cells, what they saw was these little round holes in the surface of the membrane and a tube that went in. So this is actually part of the membrane of the muscle cell. So the action potential moves down into that tubule, and it reaches sarcoplasmic reticulum here. Sarcoplasmic reticulum is a storage site for calcium. It's modified endoplasmic reticulum, membrane bound, and it has calcium pumps that are pumping the calcium in. So when the action potential gets down here, that opens voltage-gated calcium channels and allows calcium out. And so calcium gets into the sarcomeres, binds to troponin, and causes muscle contraction. Okay, so calcium is coming out of the sarcomere through voltage-gated calcium channels. Why calcium channels? Why not calcium pumps? Do you want this to be fast? So which is faster? Channels. Okay. So if you want it to be fast, channels. Okay? And this is one of the things I want you doing when you're practicing this, is thinking about the logic here. If you just memorize what's going on, if you get it backward, you have no way to notice that you got it backwards. But if you think about the logic, if you want muscle contraction to happen quickly, right? If there's a tiger in the corner of the room here, I don't want to have to wait for pumps to move calcium. I want to just open channels. Once the channel's open, that's relatively fast. Calcium comes in, I start running. Okay? And Mike gets eaten. Okay? Emma?
When the T-tubules, like when the action potential gets down to the base of like the T-tubule where the sarcoplasmic reticulum is, what exactly happens right at that moment to open those calcium channels?
So those calcium channels are voltage gated, and that voltage gate opens up from the sodium coming in and lets out the calcium. Okay? So these are voltage gated calcium channels down here. So voltage gated calcium channels. I'm oversimplifying slightly, but you don't need the additional details. Not yet. Okay, make sense? Okay, so muscle contraction then continues until you stop having action potentials and the calcium pumps have enough time to move the calcium out of the cytoplasm back into the sarcoplasmic reticulum. So calcium pumps are always moving calcium out of cytoplasm, either outside the cell or out of the cytoplasm into sarcoplasmic reticulum. Okay, so let's run through these events and number them. So we've got 1, an action potential starts. 2 gets down to the end of the axon. 3, we open up vesicles. 4, the neurotransmitter gets out. 5, open an ionotropic receptor and let sodium in. 6, open voltage-gated sodium channels. 7, spread an AP, an action potential. 8, action potential gets down deep into the T-tubules. 9, opens voltage-gated sodium channels. 10, calcium gets into the sarcomeres, binds to troponin, and starts contraction. And after time, slower process, the calcium pumps move the calcium. So muscle contraction is fast to start, but it's slower to end because you have to wait for the calcium pumps. Okay? Okay, think through all that. Close your eyes, see if you can get through the whole thing. Okay, check with your neighbor on any questions you have, anything you're not certain about, and then I'll take questions. Okay, Meg and Sarah. So the question is, how organized are the sarcomeres in relation to the T-tubules and sarcoplasmic reticulum? So if we had a muscle cell across the front of this room, what you would see is sarcomere attached to sarcomere attached to sarcomere all the way along. And T-tubules would be going in between some of the sarcomeres. So there might be one in front here, and there might be one in back here. Okay? And sarcoplasmic reticulum would be surrounding groups of sarcomeres. Okay? So each sarcomere is a long line attached end to end to end with calcium source nearby, the endoplasmic reticulum, and T-tubules coming down in between them all. Okay, Meg? So from above, we'd see T-tubules scattered all over. Okay, does that make sense? Okay, and each sarcomere would have sarcoplasmic reticulum around it. Okay, I saw some more hands somewhere. I thought I saw more hands. Okay, then question from the homework. How would muscle contraction be different if a skeletal muscle cell expressed more calcium pumps than normal? More calcium pumps. Okay, so tell your neighbor, more pumps. Okay, so here's the quick sketch I would be using. And I'd be sketching pumps are using ATP to move calcium in to the sarcoplasmic reticulum. Channels are allowing the calcium out. Okay, so with more calcium pumps than normal, we'd add okay, so what's different? Simeon? Okay, so a little less calcium is available to bind troponin. So if there's enough of them, you might actually expect a slightly lower calcium outside. So a little less, so that would mean what? Contraction would be, okay, slower and weaker. Okay, so you might have a little bit slower. That effect would be pretty small. You'd need a lot of calcium pumps because they're so much slower. Okay, but that's one. What's another one? Okay, so quicker for the contraction to stop. So contraction halts earlier. So that would be the biggest effect. Okay, you're taking the calcium out of the cytoplasm faster than you were before. And that's the one that would be the biggest. Okay. Simeon? Could you say contraction starts later than usual with the logic you had? Yes, very slightly later because the calcium pumps are reducing the calcium a little bit more at the beginning of that. Okay. It would be as it turns out, that would be a very subtle effect, but you guys don't have enough background in this yet to know that. But okay. Okay, so two sensible answers there. Okay, other questions? Okay, what if there were no T-tubules? No T-tubules. Okay, no T-tubules, you're going to get no action potential. So the only calcium you're going to get is calcium near the surface. So that's definitely going to make muscle contraction weaker because you won't get enough calcium into the sarcomeres deep in the cell. And it's also going to be slower. You're not going to get as much force applied as quickly. Okay? Okay, in the notes on page 5, I have a number of additional questions that are these sorts of questions, and they are the sorts of questions that show up on the exam occasionally. So on page 5, you can take a look at them and think about those. I want to just mention that there's more than one type of muscle cell. You can use the molecular toolkit to develop all sorts of different cells. Our skeletal muscle cells are really big, and they're what power our movement, so they need to be fast, and you need to get calcium into the sarcomeres really quickly. So because they're big cells, you've got to have T-tubules. But some muscle cells, some skeletal muscle cells are much smaller. And if they're smaller, and the calcium can get all the way through the cytoplasm without having to go a really long distance, then you can get away with fewer or even no T-tubules. So really thin muscle cells don't need T-tubules. There are other ways to regulate contraction. So some muscle cells don't have action potentials. Instead, neurotransmitter just opens calcium channels and lets the calcium in directly. But that means you need neurotransmitter all along the muscle cell. Okay, so depending upon the cells, depending upon the organisms, there's different ways to organize all of this. We're going to use skeletal muscle as a model for how things work. And in general, they're all calcium regulated. So they all involve some sort of calcium binding. You have some muscle cells that are depolarized in part by stretching. So in the gut, for example, your intestinal muscle cells react to being stretched from a large amount of food or intestinal gas. Okay? They react to being stretched by trying to contract. That helps to prevent them from stretching too much and risk bursting, not something you want your intestines to do. So as they become more stretched, they react by compressing, trying to move the food or gas or whatever it is along, causing potentially embarrassing moments. And also potentially causing cramps if they can't. So when those muscle cells try to contract, but they can't because there's nowhere for that material to go, there's just too much packed in there, that really tight muscle contraction can block blood flow. And reduced blood flow then starts to cause pain. So that's some of the pain of intestinal cramps. So the next time, Thanksgiving's coming up, the next time you've eaten too much and you're feeling that pain in stomach or intestine, I want you imagining this process. Stretch in your intestinal muscle cells, opening stretch channels, which then in turn allows sodium in, which opens voltage-gated calcium channels, which causes this intense muscle contraction. So I want you going, oh, this hurts, but I know why. Okay? Another one of my jokes falls utterly flat. Okay, I want to back up to something I didn't quite get to. I want to talk really briefly about another aspect of integument, since that's part of what we're doing here. And talk about color change. Now, pigments, so animals can have pigment that is just fixed. They produce something that absorbs some wavelengths, reflects others, and you get a color. And they can just put that in cells. They're called chromatophores. Chromato for color. Okay, chromatophores. Just put those in skin cells. That's what we do. So our hair color, our skin color, it's just melanin. in skin cells. Somewhat adjustable in response to ultraviolet. There's a balance between the need to get some ultraviolet rays to make vitamin D, but there's a risk from too much, breaks down folate, potentially causes cancer, and so people living in different environments for long enough wind up with different skin colors very predictably. The more fun part is animals that can change color, and this, in theory, could be done with genetic engineering to humans. Think about that for a moment. You could change colors like a squid or an octopus. Okay, how do you do that? Well, the way you do that is you put the pigment granules along tracks of microtubules So put them along tracks of microtubules, and then you can either spread them out, in which case there's this big patch of skin wherever that cell is, and it's red. Or you can move them all to the middle, and that cell is mostly transparent. You have one dense little spot, one tiny little spot of red, potentially so much that it actually just looks black or gray. On the other hand, if you move all those pigment granules out, the whole cell turns red, and that patch of skin is red. Okay? That changes color over minutes. So this is not fast. But it does things, for example, like allow a flounder to settle on pebbles on the seafloor and change color to match the pebbles and stuff below. Have any of you seen photographs of a flounder on a chessboard, on a checkerboard? Anybody? Some of you have. You might want to look it up. You can turn a flounder. It's not a very good checkerboard, but it can pick up a checkerboard pattern if you put it on a checkerboard. It takes some time. It's not a fast process. Squid and octopus do this really quickly. They do it by having a cell that has all this red everywhere, and then smooth muscle that can stretch it out. So think of a little sphere of red, attach smooth muscle all the way around the edge, and pull it out, and you turn it into a red pancake. Okay? And that's how squid are getting those incredibly rapid color changes. They're actually using muscle to do it. And it's worth watching. If you haven't seen a video, it's worth watching a video of these really rapid color changes. And it's also worth thinking about whether, if you had the chance, would you genetically engineer yourself to be able to do that? Think about it. You could just sit here in class right now and do sinister clown makeup at me. And you would be able to turn it off. That's the nice thing about it. You can make it come and go. Okay, so I put copies of these images on there if you want. I wanted to point out, here is how your major skeletal muscles work. Animals also can put inhibitory neurons on them. So you can inhibit muscles either by inhibiting the motor neurons, or you can inhibit them directly. You mostly don't inhibit them directly, but many invertebrates have a neuron that inhibits muscle cells. You have smooth muscle cells. Smooth muscle cells are smaller, but still long and slender. And they sometimes produce action potentials and pass them from cell to cell through gap junctions. Okay, so here we have action potentials that are traveling through gap junctions over an entire patch. So you contract when you use neurons to contract your stomach muscles, your intestinal muscles. You contract entire patches, small patches of them at a time. Longitudinal or circular muscle to move material through your gut. You use them for your eyes. Some of the smallest muscles you have control the lens shape in your eye to adjust lens shape. And the actin and myosin is arranged a little bit differently in muscle cells. It doesn't have to be all aligned in a perfect straight line like it is in skeletal muscle cells. In smooth muscle cells, it crisscrosses. And so when contraction happens, the whole cell shortens and sort of bulges out. Again, it has to stay fixed volume. Okay, so when it shortens, it has to get wider. This is the sort of stuff that I've put on exams. You don't need to memorize this, but I would want you to be able to think about the logic. So there are things in here that occur in known muscle cells, like adjusting the rate of calcium uptake, which is number of calcium pumps, right? And that changes the characteristics of the muscle. They have different numbers of mitochondria. So with more mitochondria, it takes longer to fatigue. They can keep making more ATP, but it takes space. If you put mitochondria in the cell, you have less space for actin and myosin. So more mitochondria, you don't fatigue, but those cells aren't as strong either. So if you want maximum power, you put minimum mitochondria, minimum everything except actin and myosin. You get power, but not for long. So there are all sorts of trade-offs in muscles. We talked about how tightly phosphate binds. So the top one there, myosin ATPase. Myosin is an ATPase, right? It uses ATP to change. So myosin ATPase, how fast or slow it is affects how fast or slow contraction occurs, but also how expensive that muscle is. The duration of twitches of each contraction is partly related to how many calcium pumps there are. Lots of capillaries around the cell. You get a lot more oxygen, more glucose replacement. You take away waste products, okay? But you have less space for the muscle, for the actin and myosin. And so more blood vessels means your muscles will keep contracting longer, but not for as strongly. Okay, so let's take this and move now to the macro scale. And I want you thinking about how joints work. So that's another video you watched. And I hope this makes sense to you. If this is your arm, okay? And apparently you have 4 fingers and no thumb. There you go. You always need to know where pivot point is for these particular types of joints. In a moment, I'll comment on some other types of joints. But a pivot point, if you pull up on this, you're going to make the muscle a bit shorter and a bit fatter. And your arm is going to move. So that's your biceps, the flexor, and muscles ordinarily have an opposing muscle, an extensor. Remember, an extensor is opening up, making a larger angle. A flexor is making a narrower, smaller angle. So if you pulled up on the triceps, that would move your arm in the opposite direction. Okay? So hopefully that makes sense to you. I also want you thinking about, does it matter where you attach it? So what if you attach the muscle way out here? So change the attachment, get the same amount of shortening, and you have less distance traveled. So attachment points that are far from the pivot are slower. They also have a mechanical disadvantage. So you can't get as much power, so you get more, they have a mechanical advantage if they're further away, so you get a little bit more power. So animals that use their forearms for digging, instead of the biceps attaching really close to the joint, the biceps attaches further out on the forearm, and they get more power. It is slower, but they get more power. Animals that are runners or need really fast motion, the muscles attach very close to the pivot point, and you get fast movement. Okay. What if it's an exoskeleton? For an exoskeleton, again, you always need to think about where the pivot is and what the muscle is going to do. So if this was a pivot, and we had a muscle here, is that a flexor? Or extensor? Okay, that's going to pull here when it shortens. And the most this can do is get pretty straight the way I've drawn it. So you always need to know the pivot point. Started to draw this a different way, but it would have required a different pivot. Okay, if we shorten this one, and that's still our pivot point, that's going to pull down. It's going to be a flexor. Whereas our first one, A, is going to be an opposing muscle to that one, an extensor. Simeon?
So I think it's easier to tell endoskeletons from exoskeletons. How do we know, like, for that pivot point, which angle?
Yeah, how do you know which angle you're talking about? On the exam question, I will try to make it clear. Because if on this one, this could do enough to make it shorten the angle this way, then it becomes harder. Okay. But if this muscle is bending down here, so if the range of motion, so if we decided the range of motion is here to here, okay, if that's as far as it can go, then the extreme is about 180 degrees. And this muscle is an extensor that will get it up to there. If it can bend all the way down here, if this one can shorten enough to bend it all the way down here, then that's a flexor. It's making a narrower angle. Make sense? Okay. And you need to know the pivot points always to be able to figure them out, no matter what they are. Okay, that's for joints that have a single pivot point. There are joints that are sliding. There are joints that rotate, ball joints. So they get more complicated, and so the science of anatomy winds up being, and of muscle movement, winds up being a lot more complex than just these movements. But these movements are the starting point. Okay, and I don't want to have you focus on a whole bunch of different joints that we aren't going to need to make designs for neural circuits and things for movement. Okay? Any questions about these? Any other questions? Okay, some other comments on skeletons. So endoskeletons, it turns out one can grow and still use them. You can have a long, tough part, a hard end, and have a thin little region where growth is taking place. And as long as you don't mess that up too violently, it's still a rigid bone that you can use, which enabled you to grow up slowly, to increase in size slowly, and be able to move that whole time and even do fairly rough stuff. Okay, because you just had a thin point at each end of your long bones with a bony cap over it, okay, where growth is taking place. And there's just thin, slow growth with enough structural support so you can still use your bones. It's hard to do that with an exoskeleton because the outer part is the outer part of your body. So how do you keep that growing when you have no cells, no live cells over that surface? So exoskeletons have to be molted. And this is why arthropods wind up getting to a certain size, and then they degrade and reabsorb the lower part of their exoskeleton, hang on to those resources, crawl out, pop open their exoskeleton and crawl out, and then use the resources plus some that they've already stored to make a new, bigger exoskeleton, and then slowly grow into that and repeat. It's very expensive, and there's a period of high vulnerability right after that molting has taken place. So exoskeletons have some advantages. You have a hard outer surface all over, but also disadvantages. Exoskeletons have another problem that as you get bigger and bigger and bigger, remember the problem we talked about before, that as an animal increases by a factor of 2 in all dimensions, it is now 8 times more massive, but its legs have only increased by a factor of 2 times 2, 4. So it's 8 times more massive, but only on 4 times the support. And so as animals get bigger, like us, you need thicker and thicker legs. We have comparatively thick legs. If we were the size of a mouse, our legs could be, probably something like this big around. Just imagine yourself or me standing up here on little stick legs like that, okay? Because the mass per cross-sectional area, per unit cross-sectional area is smaller. Well, the same problem happens to an animal with an exoskeleton. If it increases by a factor of 2 in each direction, it has 8 times the mass, but its exoskeleton has actually only gotten 2 times thicker. So it is weaker in proportion. Well, if it's going to increase its protection and increase its ability to support at every point the internal mass, it needs to increase its exoskeleton by a factor of 8. Okay? And as the animal gets bigger and bigger and bigger, it needs a thicker and thicker exoskeleton. So at some point, it can't make an exoskeleton any bigger. So one of the constraints on size, fortunately for us, of arthropods is that as they get really big, their exoskeletons get so massive that it's hard for them to move them around, and it's very expensive to make them. This is why there are no lobsters the size of tigers roaming around the US. Okay? We don't have to worry about lobsters the size of tigers because Lobsters that size would need such thick exoskeletons to protect them even from falls. So even falling would crack them. Okay. Tiny mice don't have to worry much about broken bones. They can fall almost any distance. Their bones don't break. You can fall and break a bone. I know this from experience because I've done it. But not in class, so no worries. Okay, last bit of what I want to talk about today is moving and staying in place. So we've talked about muscle movement, all the way from the molecular out to movement of joints. Okay? And the last piece is, how do you actually move against the substrate? So if I was on ice and I tried to move, I'd be having a lot of trouble. Every time I try to push myself forward, my feet would tend to slide out from under me. So on a really slippery surface, it's hard to move. We rely on friction forces, gravity pushing our body down, friction with the ground, so that when I push forward or push back against the ground, I lean forward. So friction forces for terrestrial animals and for massive aquatic animals, so friction forces are very important. Our movement is basically a series of controlled falls. So we push forward, overbalance, fall. Push forward, overbalance, fall. So it's a series of controlled falls, and we use friction forces to stay in place. Others, especially for smaller animals, friction forces are insufficient. In water, friction forces are often hopelessly insufficient. Glues, we've already talked about. So animals glue themselves in place with temporary glues. They use things like mucus. So many proteins, some of them in mucus, use glue. Some terrestrial animals, like aphids, use a thin film of water and hydrogen bonding to stick to things. So really small animals can even use water as a glue. Suction is another one. So many, many animals have independently evolved suction cups to hold themselves in place. All that takes is having a disc shape that can seal itself to the substrate, pull up on the center, you create an area of negative pressure, lower pressure, and the higher pressure all around will push that suction cup onto the surface. Van der Waals forces, so weak molecular attraction. The famous examples are geckos. Their toe pads split into many, many split, so that they have, I don't know what it is, thousands, tens of thousands of tips. And when they push their feet against the surface, all these tiny little fibers fit into all the little cracks and attach them by weak molecular force, but over a large surface area. Okay? And so they're able to run around on walls, on surface, upside down, and so on. And then charge interactions, so uneven charge distribution on sugars. So some animals are using surfaces that have uneven charge distributions, and against another surface, put those down, and the charges, if they can arrange themselves to match opposite charges, they'll stick using ionic bonding or hydrogen bonding. So once they're stuck in places, movement, so our controlled falls, running or walking on legs. Some animals attach themselves to a location with protrusions like setae or a grip, and then detach, pull themselves forward, pull longitudinal muscles, so worms, movement of worms. Some animals push themselves against substrates, so salamanders walking have a movement like this, and they're constantly putting out their leg, attaching it or hooking it over something, or using friction forces and doing this. And then swimming or flying, pushing a fluid or a gas away from your body. So push a fluid or a gas away, and that imparts momentum to you to go in the opposite direction. And then the recovery stroke has to minimize viscous interactions with the water. Okay, next time we'll start talking about excretion. I do want you to remember Reynolds numbers. So at the end of this chapter, the posted notes, I have a review of Reynolds numbers and the ratio of inertial forces to viscous forces.